Hello, this is Gina Piscatelli with the second lecture for the immune system. And this lecture focuses on one branch of adaptive immunity called humoral immunity, or you can call it antibody-mediated immunity. The cartoon on this introductory slide shows some green rod-shaped bacteria that are trying to outrun or avoid those yellow antibodies made by a plasma cell. The plasma cell is a type of B lymphocyte that we'll talk about in a second. So humoral immunity involves antibodies as the effectors or the ammunition of humoral immunity. And there are two sources of antibodies. They're proteins found on the membrane of B lymphocytes or secreted in soluble form by plasma cells. And as I mentioned previously, plasma cells are formed from or differentiated from a B lymphocyte, and we're going to talk about that next. Before we get into the details of how those antibodies work and how humoral immunity protects us, we're going to look here at how B lymphocytes develop and where they're located. For example, we'll discuss how those plasma cells I just mentioned form briefly. So this is the life cycle of B lymphocytes, or called B cells. They originate, like all white blood cells, in the red bone marrow, and that's where they mature. They're named B cells not so much for bone marrow as because they were first identified in the bursa of a chicken. So it doesn't matter. A bursa is like a protective fluid-filled sac. It's got synovial fluid in it. You have one underneath your patella. Anyway, in the bursa of a chicken, these B lymphocytes were first identified, so they're called B cells. But anyway, they do mature in the red bone marrow from stem cells. And those stem cells divide, and a product of the division matures by a process called selection. And this occurs for B lymphocytes in the bone marrow. And during this maturation or selection process, they become what is called immunocompetent and self-tolerant. And for now, we're just going to say they're mature, and I'll explain what those two terms mean in just a second. So after the B cells mature in the red bone marrow, they're called naive B cells because they haven't really yet encountered an antigen. They've just been made. But once they have been made, these naive B lymphocytes can travel in the bloodstream from the red bone marrow to secondary lymphatic organs where they reside. That's where they live. Not that you can't find them in the plasma, you do, but many of them live in secondary lymphatic organs like lymph nodes. And this is where they first encounter foreign antigens. And in truth, most often this encounter does happen in a lymph node, but there are other secondary lymphatic organs that we've already studied, like the spleen and the tonsils, Peyer's patches in the small intestine, lots of secondary lymphatic tissues and organs. Well, once a B cell recognizes and interacts with this foreign antigen in the secondary lymphatic organ, it proliferates. And that means it divides, and the daughter cells divide, and then those daughter cells divide. So you get this huge production of B lymphocytes that are clones of the original, and those clones recognize the same antigen. Some of those clones will differentiate into what are called plasma cells, and those are the purple cells shown at the very bottom, right here. But some of those cells, clone cells, become memory cells. And these memory B cells and plasma cells, as well as the antibodies made by the plasma cells, then can cycle from the secondary lymphatic organ either into lymphatic vessels or blood vessels, and they'll often circulate between the two. So you can find them in both the blood and the lymphatic fluid. So I want to mention a little bit more about this process of maturation, specifically the terms immunocompetent and self-tolerant, what those mean, because they're important for both branches of adaptive immunity. The process of lymphocyte maturation and selection is really better understood in T lymphocyte maturation, but we're going to use what's known for T cells while discussing B cells in the bone marrow, because I guess a similar process happens, even though the literature is a little bit more vague. So during the development of B lymphocytes, the only ones that survive and leave the bone marrow as fully mature B lymphocytes, naive lymphocytes, are those that become immunocompetent and self-tolerant. And what immunocompetent means is that one specific B lymphocyte, one particular cell, makes antibodies, multiple antibodies, but all of those antibodies recognize the exact same antigen. So a B lymphocyte becomes immunocompetent when all of its antibodies recognize just this one particular antigen. This is determined, what antibodies the B lymphocyte makes is determined by the nucleotide sequence in the genes that code for antibodies. So there's a set of genes in the B cell, in the nucleus of the B cell, and there's a certain gene that codes for antibodies. Actually, there's more than one involved, but a gene that codes for antibodies. So essentially, the types of antibodies your B cells make, all of your B lymphocytes, is largely determined by the genes that you've inherited. However, these genes can be rearranged when a B lymphocyte divides, and that of course happens in the bone marrow when stem cells divide. So when genes are rearranged, the genes code for a different antibody, and this gives our immune system multiple types of B lymphocytes and the ability to recognize almost an unlimited number of antigens, few of which we will ever encounter. So that's immunocompetence. The other component of becoming mature is self-tolerance. And this means that a B lymphocyte during the maturation process is exposed to our normal self-antigens on our body cells. And it has to learn that those are not foreign antigens and not to make antibodies against those self-antigens. Any lymphocyte that does make antibodies that recognize our own cellular antigens is destroyed. Okay, so back to antibodies. Let's look at the two forms of antibodies. Some are secreted by a plasma cell, so we would call them free or soluble, and they're blue here. And some antibodies are actually made by a B cell and located on the surface, a B lymphocyte. So some B lymphocytes put the antibody right on the cell surface like a membrane-bound protein. Both types of antibodies can recognize and bind always to a foreign antigen, but the foreign antigen could either be free or soluble, like a toxin or a virus, or, and that's true in both cases. Here's a soluble antigen on the right, can bind to that type of antibody or a free antibody. Or the antibodies can bind to an antigen that's on the surface of a bacterial cell. And here's a bacteria on the left that has foreign antigens on the surface, and a free or soluble antibody can bind to it. And on the right, we have a B cell that has antibodies attached to its cell surface, and it can also recognize foreign antigens on a bacterial cell. So the antigen doesn't have to be by itself, it can be on a foreign cell. It turns out, though, that antibodies generally do not bind to infected host cells. So if a virus, we'll pretend this soluble antigen here is a virus, if a virus enters one of your body cells and then that antigen is displayed on its surface, then the antibody doesn't bind to it. And that would be for the cell-mediated branch, not for the antibody-mediated branch of adaptive immunity. Okay, let's look at the structure of antibodies a little bit. Antibodies are proteins that are called immunoglobulins, so you'll often see the abbreviation Ig when talking about an antibody, Ig for immunoglobulin. And the basic structure of all antibodies consists of 4 polypeptide chains. And here we have 2 that are light chains and they're purple. And then we have 2 heavy chains that are blue. And they're linked together with disulfide bonds. And they're shown here in yellow. The disulfide bonds are yellow. So these disulfide bonds create loops in both chains, as well as link a light chain to a heavy chain and link the 2 heavy chains together. So the 2 heavy chains are longer, so technically I guess they weigh more. And the other 2 chains are shorter. They're called light chains. And at the tips of the whole Y structure, so this is like a Y-shaped molecule, at each tip there's an antigen binding site. So there's 2 antigen binding sites on this one antibody. And the antigen binding site is located in what we call the variable region. And the variable region in this particular picture is darker, like the color is darker. It's not faded out like the rest. And this is the region that varies depending on what antigen it recognizes. So that's what gives the antibody its specificity. At the very end of the heavy chains, I guess the base, if you want to call it, here at the base of the Y, is the stem region. It's this stem region that determines the class that an antibody belongs to. So the stem will have a sequence that differs, a polypeptide sequence that differs by the class of antibody. And there are 5 different classes that we're going to look at, and they function just a little bit differently during our immune response. So the molecule as shown here, the antibody shown here, is Y-shaped, and it's also referred to as a monomer. Okay, so this is the basic structure, a monomer of an antibody. Now let's look at the other classes. There's 5 different classes, as I mentioned, and IgM is made up of 5 different monomers or 5 different Ys. So there's a Y, there's a Y, etc. So we call this a pentamer sometimes. So that's IgM, and it's secreted exclusively by plasma cells. You won't find those on the surface of B lymphocytes. And I guess what's most important clinically is that this is the first type of antibody that's secreted when you're first exposed to a novel antigen, something that your body hasn't seen before. Which is a little bit strange now that I think about it, because plasma cells are usually derived after you've been exposed. So I don't really know the answer to that. Presumably there are some plasma cells that are created, and IgM, I don't know, it's confusing. Then there's IgA, which is a dimer, and this type of antibody is found in saliva, sweat, intestinal juice. It's also found in mother's milk, breast milk. And the rest are all monomers. So IgD, IgG, and IgE, those are all monomers. And we find IgD on the surface of B lymphocytes. We find IgG in the plasma, most abundant antibody in the plasma. And it's secreted in large quantities during the secondary response by plasma cells once again, because those are the ones that secrete the antibodies. And IgE is really interesting because it is really important for inflammation and allergic reactions. This stem of the IgE can bind to mast cells and basophils, causing histamine release. And we'll talk about how that affects the immune response in a little bit. So those are all the different types of antibodies. Now, what do they do? As a rule, antibodies don't recognize infected host cells. Instead, they recognize either foreign cells or foreign molecules. Those molecules are antigens, might be on a cell, they might be free. And what they do includes neutralization. That means they bind to whatever's foreign, like this yellow thing, which is probably a virus, and masks its harmful effects, so it can't do whatever it's supposed to do. Like the effects of a toxin might cause a fever, right? Or microbial cells that want to proliferate, divide. But the antibodies don't kill anything, they just mark them for destruction that other phagocytes will perform. Another thing that antibodies do is called agglutination. And in this case, the antigen is on a foreign cell, because agglutination means the clumping of cells. So the antibody has bound to a cell, and because the stems can interact, well, IgM is already a pentamer, but the stems can still interact, and you get clumping together of all those foreign cells. And this is what happens like we did in our blood typing, or it happens with a transfusion reaction. So that's agglutination. The other thing they can do is precipitate a toxin. So here we've got 3 different antibodies binding to just one antigen molecule. And therefore now the antigen isn't so soluble. It can't move around as easily in our body fluids, which is good because it's like localizing the toxin. Now there's 2 other ways that antibodies can fight pathogens. One is, the second one I guess, is that they can cause cell lysis. So I said that antibodies don't kill directly, and that's still true, because how they work to elicit cell lysis, shown in this picture on the right, is that they activate complement. Remember, complement is a set of proteins normally in an inactive form, but when they're activated, the complement proteins make this pore. And so when an antibody has bound to a foreign cell, like this green cell, or in this case it's a yellow cell, the antibody binds. Complement can bind to the stem and be activated and form this membrane attack complex. So it's really complement that's doing the killing, so to speak. Okay, the third way that antibodies help us fight pathogens is to enhance phagocytosis by other leukocytes, other phagocytes. What they do, antibodies, is tag antigens for phagocytes to easily recognize. So in the picture on the left, the antibodies happen to be membrane bound. But that isn't how it started. How it started was that this green pathogen entered the body and some soluble antibodies, they're shown in blue, bound to it with the stems exposed. But now that phagocyte, which is purple, binds to the stems. So I guess it's sort of membrane bound and can more readily engulf that green foreign pathogen. Another thing that causes phagocytosis, of course, would be things like neutrophils and macrophages, neutrophils, etc. What else enhances phagocytosis is that antibodies, when they bind to mast cells, shown right here, a mast cell, and the antibody that's already bound to an antigen binds to a mast cell, which causes it to degranulate. Degranulation just means that these mast cells or basophils, same thing, release histamine. And that histamine causes blood vessels to dilate. And when the blood vessels dilate, now all these other phagocytes that I just named, neutrophils, macrophages, etc., eosinophils in some cases, can perform phagocytosis because they're going to actually be able to get out of the blood and go to the site of infection. So 3 different ways that antibodies fight pathogens in broad terms. They can inactivate a foreign antigen. They can indirectly cause cell lysis by activating complement. And then the last thing they do is enhance phagocytosis. But in all cases, they have to bind to the foreign antigen for any of those to work. Okay, let's look at how B lymphocytes fight pathogens. It turns out that B lymphocytes are kind of like phagocytes. They can engulf foreign antigens. And they do that because the B lymphocyte, not a plasma cell, has antibodies on its surface. Those antibodies are like receptors. And so receptor-mediated endocytosis can happen, which just means engulfing the entire antigen-antibody complex. And then once I haven't drawn an antigen on here, I guess I should. Let's do an antigen in blue. Now once engulfing happens and the antigen is on the inside, which I'm not that sophisticated with this mouse, but now the antigen is inside, the B lymphocyte processes that antigen, kind of breaks it down and puts it with some molecules called major histocompatibility complex. And these molecules are abbreviated MHC1 and MHC2. So I'll draw a MHC molecule on the surface, and then the foreign antigen sits on the outside. So at this time, just so you know, and I should just do it in white here, you know, the receptor and antigen stuff, that's all gone. Okay, once the B cell has engulfed the antibody and the antigen complex and then processed the antigen, now the antigen is displayed on the surface of the B cell with this red MHC molecule that I tried to draw. Okay, so what big deal? They've engulfed a foreign antigen, which is good because now it's not freely floating. But what's really cool is that these B lymphocytes can present this antigen MHC complex to T lymphocytes, but only a certain type of T lymphocyte, a helper T lymphocyte. So what happens when that occurs? Well, it activates the helper T cell, which really is part of the cell-mediated branch, but we're seeing some crossover here. So once the helper T cell recognizes this complex presented by the B lymphocyte, the helper T cell binds and it releases cytokines. Okay, now what? Well, the cytokines cause proliferation of that original B lymphocyte type. So you're going to get a lot more, a higher clone production, right? And the cytokines recruit other white blood cells to perform phagocytosis. So I have a picture of this in a couple of forms here so that it'll make more sense to you. So what do these B lymphocytes do themselves? First, they engulf antigens via endocytosis or receptor-mediated endocytosis. And so here you see this antigen has been engulfed. The antibody just gets reused. But the antigen goes on the surface, complexed with MHC1 or MHC2. Those are major histocompatibility complexes. We'll talk about those later. All your cells have one class. Anyway, so the B lymphocyte has both MHC1s and MHC2s. But the helper T cell that it presents this thing to, which is shown down here on the bottom left. So here's a B lymphocyte presenting that antigen that's on an MHC. When it presents it to the T helper cell, the only way the T helper cell is going to recognize it is if the antigen is on an MHC2. But regardless, the helper T cell then binds to this complex of the antigen on an MHC2, and then you might remember, I don't know, that T lymphocytes that are helper T cells are called CD4 cells. So the CD4 cell, or the CD4 molecule helps ensure that it's an MHC2. Anyway, it doesn't matter. The point is that the helper T cell binds to this B lymphocyte that's presented the antigen to it, and now the helper T cell releases cytokines. And these cytokines have funny names. Interleukins just means that one white blood cell is going to talk to another white blood cell, and there's a bunch of them, so they've got numbers. But anyway, what these cytokines or interleukins do is cause proliferation of this exact type of B lymphocyte. Some of the cytokines will actually attract, by chemotaxis, other white blood cells, like macrophages, neutrophils, basophils, natural killer cells that can, use their resistance mechanisms to fight the infection. This is a different picture which shows the same thing, but it might be a little bit clearer for you. It doesn't show the engulfing process that a B cell has to do, a receptor-mediated endocytosis, but it does show that there is a foreign antigen that's red on an MHC2 molecule being presented to a helper T cell. The helper T cell recognizes the antigen and MHC2 combination, and that causes it to release cytokines. It turns out that it's interleukin 4 that actually causes identical B lymphocytes to proliferate. But the point is, if you get a lot more B cells that recognize the same antigen, you're going to get more antibody production, and that will amplify the humoral response. So B lymphocytes do antigen presentation, which will become, we'll talk about it again when we do cell-mediated. Okay, let's look at what really happens when you are first infected with some kind of antigen, foreign antigen. We call this the primary immune response. And I'm only going to talk about the antibody-mediated branch to begin with here. So the first thing that has to happen is that the antigen has to be recognized as foreign, and an antibody will bind to that antigen. Let's say the antigen is on a B cell. The antibody is on a B cell. That B cell then engulfs the antigen by receptor-mediated endocytosis. And then we get a proliferation of B cells that can recognize that exact same antigen. Some of the cells that result from forming this clone of B cells will become plasma cells and just pump out enormous numbers of antibodies. Your book estimates 2,000 per second, but I've seen 10,000 per second, so enormous amounts of antibodies. Other B lymphocytes that result from this proliferation will become memory cells, and those memory cells are going to go live in your secondary lymphatic organs. They won't do anything right away. They won't even make antibodies, only on their surface. They won't pump out antibodies that are in secreted form. And then, of course, some of the B cells that are clones will do the antigen presentation we just talked about and interact with those helper T cells. So there's 3 steps. First, recognition of the antigen, then proliferation of identical B cells, but those products of proliferation differentiate. Some become plasma cells, some become memory cells, and some of them, I guess they're still just B lymphocytes, interact with T cells. And this is what it looks like in pictorial form here, a picture, instead of just me talking. First, you have to have antigen recognition by a naive B cell, usually, one that hasn't. It doesn't always have to be that way, but then it would be a memory cell. But anyway, so an antigen is encountered by a naive B cell. Now notice that only B lymphocytes with the appropriate or complementary antibodies will bind. So this B lymphocyte that has the green antibodies, it's the wrong sequence. The lymphocyte that has the blue antibodies, wrong sequence, can't bind to that antigen. So after binding and the formation of clones or cell proliferation, notice that we only get B lymphocytes that make the blue antibodies. So we get high numbers of B cells. And just the fact that there's 2 here doesn't really give you a feel for the fact that there's subsequent cell divisions. So you get heaps of cells. Some of them become plasma cells and secrete a large quantity of antibodies that can travel throughout your bloodstream and in your lymphatic system. But other products of this proliferation become memory cells. And those are the ones that go live in your secondary lymphatic organs. So the overall effects of the primary exposure and subsequent response by the humoral branch of the immune system is the production of massive amounts of antibodies for 4 to 5 days. And those are made by plasma cells that have a short life, but they sure make a lot of antibodies. They have an extensive endoplasmic reticulum that just makes them pump out protein like crazy, and of course ribosomes too. But then they die. But memory cells have also been formed, and they live for a very long period of time. They can live for years in a secondary lymphoid organ. It depends on the antigen and whatever else. So those memory cells can change or divide later if you're exposed and generate plasma cells within hours after you've encountered the exact same antigen for a secondary response. And during this secondary exposure, these plasma cells make even more antibody than the plasma cells from the primary response. And they do so quicker, within 2 to 3 days, it says here. Times are, just know primary response is short, secondary response is quick. So this is a pictorial representation of the secondary response that occurs. So secondary exposure. And this can occur years later. And it is elicited by a memory cell. So this memory cell makes antibodies that respond to a foreign antigen that now you're seeing for the second time. And again, you're going to get rapid proliferation. Evidently, it's even faster cell division than occurs during the primary response. And the plasma cells that are made make tons more antibodies. So not only do you get more plasma cells, but each plasma cell makes more antibodies. And again, you're going to get more memory cells. That part won't go away. So if you recall at the beginning when we were talking about the different classes of antibodies, I said that IgM antibodies are the ones that are made when you're first exposed, and that is true. I do not understand the pentamer thing, but that's okay. I don't have to understand everything, right? But IgG antibodies are made in the secondary exposure. So I'm going to point that out again as I go through this graph that shows the plasma concentration on the y-axis, plasma concentration of an antibody, a certain type or specific antibody for a particular antigen, and how that concentration changes over time. So time is in days on the x-axis. So let's first consider the first exposure to an antigen that we're going to call antigen A. And this first exposure occurs at time point 0, right there. By 10 days, I'm estimating that that's 10 days. Okay. By 10 days, the amount of antibody in the plasma that's specific for this A antigen increases tenfold. And it maxes out as well. But then note there's a second exposure to antigen A at time or day, I was going to say time point, but day 28. And now look at the quantity of antibody produced. It's like 3.5-fold times. So like 3,000 times the amount. Okay. Even if my math is wrong, which is completely likely, the amount of antibody increases drastically. So this second type of antibody that's made is IgG. But that's just the class. It's still specific to type A antigen. Okay, also notice one other thing that's kind of interesting, that if two antigens are introduced at exactly the same time point, antigen A, which you've already seen before, and antigen B, which the body has not seen before, you will not get this high response for antibodies against antigen B because it's the first exposure for B. So there will be different plasma cells that make antibodies against B than against A, and it's still in its primary exposure. So that's why the number's lower. Okay, that is all for the humoral branch of adaptive immunity. Thank you very much for listening.